Hello. Very good morning to the student who have joined. Can you let me know in the chat box if my voice is audible to you? Kindly confirm. Okay, thank you so much. And now can you please let me know if the presentation is visible? If you can see the PowerPoint presentation clearly, can you let me know? Is the presentation visible? Okay. Thanks for confirming these things. So we will start in 5 minutes. Okay. Let other students join. Meanwhile, please let me know if you have any doubts or any queries from our previous class. If there is any, you can let me know now. Okay. We will start our session in 5 minutes. So kindly wait till then. I hope my voice is audible to you and you can see the presentation and the video clearly. If not, please let me know. Like previous class, I have to mute your microphones and cameras so that I can record the session without any problem. But the chat box is always open, as I have said earlier also. So if you have anything you want to let me know, please write in the chat box. Okay. Just wait. We will start in shortly. Let other students join. I hope I am audible, right? If there is any issue with my audio or anything, please let me know. We will start in at 10 a.m. So please be there. So is there any query from previous class? In the last class, what we have discussed about the amino acids. We have discussed about the shorthand notations of the amino acids, how to face questions related to these things if the shorthand notations are given. I have discussed, and we have discussed a little bit about the how to solve or how to calculate the isoelectric points. We have discussed about other things of related to our protein and the structures of the protein and other biological compounds. Okay, so Nirmal, do you have any query? You have raised your hand. If you have your doubt, please let me know in the chat box. Okay. Okay, I think there is no doubt. So yeah, that I was telling. We have discussed little bit about the the other biomolecules like lipids, the or the prostaglandins and these things we have discussed yes in our previous classes. So today we will discuss about basically we will discuss about the deep protection chemistry a little bit about and how that particular thing is incorporated into our protein and our peptides and all these things. Okay, just let us wait for 2 more minutes and then let us start with the introductions. Meanwhile, in between the class, if you have any problem or any doubt, kindly let me know. Okay, so it is 10 a.m. So I think I should start the class by introducing myself and let other students join in time. So let us start with the introductions. I hope the slides are visible. If not, please let me know. Okay. So, very good morning to all the students who have joined. My name is Mousumi Deb. I am the PMRF Fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL SWAYAM course of Organic Chemistry in Biology and Drug Development. Today, it is our week 2 live session. So, yes, in the previous class, which we had on our first, I think on 25th of July, that was our week 1 live session. And so, every week the live sessions are scheduled on Friday from 6 to 8 p.m. But due to some unavoidable circumstances, I couldn't take the class on this Friday, which was earlier scheduled on 1st August. So today it is our week 2 live session. So sometimes it may happen that the classes are rescheduled, but you will be notified well earlier regarding this all this rescheduling so that there is no issue. And anyways, I am recording the session so the students who cannot attend the class on that day, they can check the recording later also. So before starting the session, let me just point out few things. So there will be a problem solving sessions every week where I will try to solve the particular questions related to a week's content. Okay, and these sessions are scheduled on Friday from 8 to 6 p.m. As I have said, that today is a rescheduled class due to some issues. I couldn't take the class on Friday, but the next class will be on Friday, which is I think on 8th August. So that will be our third session. Will be happening on that particular day in this time. So the sessions are live on Google Meet platform. This is the link, and this link will remain same for our entire course. You can use this link to join the Friday class, which is scheduled from 8 p.m. to 6 p.m. Okay, you can write down this particular link, or you will find it on the SWAM code page also. So the important thing is, if there is any network disruptions happen from my side, please do not leave the session. Okay, I will try to rejoin as soon as possible. I am recording the session, so if yours in from your side the network is unstable and you have to leave the session for some time, you do not have to worry. This call is open, so you can rejoin as whenever you want. You can, or whenever it is possible, you can rejoin the class. Okay, and I am recording the session, so you don't have to worry about missing anything, because this recording will be shared to you after the class. So you can just go through the entire recording to find the things which you have missed. Okay, so during the session you can post your doubts, your queries, everything in the chat box. I have said it earlier also that I am recording the session, and since I am recording the session, I want it to be very seamless, and that is why I have muted your microphones, and I have muted your cameras also, so I can just record the session without any issue. But the chat box is always open. I am monitoring the chat box each and every time. So you have any query, your doubt, anything, you can just drop an question in the chat box. Okay, the chat box will also use to during our question answer session. Okay, as a medium of your answering the questions. Okay, after the class, if the doubt persists, you can drop an email to me. I will clear your doubts through email also, and if it is not possible to do it by email, I will try to solve your doubt in the upcoming classes. Okay. So as I have mentioned already, the presentation and class recording will be shared to you. Now, last day, many students has asked me that where to find this recording, and also where to find the PPT which I am showing today, and also the PPT which I have showed you yesterday in the last class. Okay, so what you have to do is you have to go to the SWAM portal. Okay, this is the announcement page of the SWAM portal, where you will find, after login in, you will find there is a left panel. Okay, here is also your class reminders, and also everything is posted, so you can just check it from here also. And you will see there is a weekly course updates, like the lectures and also the quiz, feedback form, everything is given here. In this left panel at the end, you will find this thing, problem solving session July 2025. If you click on that particular thing, you will find there is a survey which you can fill. Okay, there is a Google form, so which you have to fill, and also you will find a problem solving session recording. If you click on the problem solving session recording, you will be redirected to a page which looks like this. You will find the YouTube link of each and every class. Okay, this is the link for the week 1 class which we have taken. I have taken earlier also earlier, and there you will find a Google Drive link. If you click on that particular link, you will be redirected to a page which looks like this. This will have the particular PPT. Okay, now which I have shown in the particular class. So we have already done our first class. So week 1 PPT is given here, and after today's class, I will upload the week 2 PPT in that particular Google Drive itself. Okay, so these are the things which you have to keep in your mind during the for the sessions. Okay, so let us start with our today's session. Meanwhile, I think most students will join in upcoming time. So today it is our, as I have already mentioned, that today it is our week 2 live session. So in our week 2 live session, what we will do, we will discuss the week 2 course content. Okay, the content which is uploaded in last week, I think in the week 2 course content has been uploaded. So today we will discuss week 2 course content. So like in every live session, what I do is I show you around 20 MCQ questions. All questions are related to that particular week's course content, and after you try to answer the question, I will show you the correct answer of a particular question. And also after that, what we will do is we will discuss that particular question in detail, so that we can can understand the particular topic very well. And also, if the subjective questions are given sometime in during a final examination, you can easily answer that. Okay, so that's how we proceed in our live sessions. So I have introduced myself earlier also. So my name is Mousumi Deb and I'm a PMRF Fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. So let us start with our group week 2 live session. So the week 2 course content for this course, organic chemistry and biology and drug development, are these. So these are the method of determination of amino acid sequence, primary structure of the polypeptide or protein, selective peptide bond cleavage, enzymatic and non-enzymatic methods, peptide synthesis, protecting groups for amine and carboxyl functionality, peptide synthesis, protection, coupling, deprotection methods, and recent developments of the coupling agents like Merriffield's method of solid phase peptide synthesis. Okay, so this is the topics which we are going to cover in today's class. So in last class we have discussed about amino acids. We have discussed about little bit about how the amide bond formation happens, the peptide bond formation happens, and so on. And so today what we will discuss that what is the amino acid sequence, how to basically identify what is the particular, if the particular given chain is given, so how to identify which amino acid is where or which amino acid is bonded with other amino acid, and so on. So these things which we will discuss in today's class. I hope my voice is audible. If there is any issue, please let me know. Okay, so let us start with the first question of today's session. So the first question of today's session says that which of the following statements is false? Whenever there is a true false type of question or there is a correct incorrect type of question, please read all the options or statements very carefully, because most of the students, I find that they tend to make a mistake in this type of questions, because this type of question sometimes can get tricky. Okay, so which of the following statements is false? Your options are: number A, the enzymes are called artificial scissors for peptide bond cleavage. Number B, proteases prefer small peptides. Number C, peptidases can be called proteases. And number D, an enzyme with broad specificity is not suitable for peptide degradation. So which of the following statements is false? Let me know in the chat box A, B, C or D, which of the following statement you think is false one. Statement number A, the enzymes are called artificial scissors for peptide bond cleavage. Number B, proteases prefer small peptides. Number C, peptidases can be called proteases. And number D, an enzyme with broad specificity is not suitable for peptide degradation. So during the session, the questions I am showing you, so these are not marked, so you won't have to worry about any kind of negative marking or any kind of marks, okay, because I am not going to mark you for the questions. So you can answer whatever you think is the correct one. It is basically a practice session, so there is no issue with the wrong answers. But I want all of you to participate and answer, otherwise I won't understand if you are following the class or not. Let me wait for 2 minutes. Which of the following statements is false? Enzymes are called artificial scissors for peptide bond cleavage. Number A. Number B. Proteases prefer small peptides. Number C. Peptidases can be called proteases. And number D. The enzyme with broad specificity is not suitable for peptide degradation. So, should I check the chat box now? Or I will wait for a minute? Okay, let me check the chat box. Let me check if there is any answer for this particular question. Okay, I have got two answers. Both are saying it is number D. Okay. So the correct answer is actually number B, which says that proteases prefer small peptides. Okay. So basically, enzymes, they are called artificial scissors for the peptide bond cleavage. So because the scissors, what they do, they cut the things basically, and enzymes, they do also the same thing. They cleave the peptide bonds. Okay. These two I will discuss later. So enzyme with a broad specificity is not suitable for peptide degradation. This statement is not false. This statement is true. As enzyme, if it has the broad specificity, it is not suitable for a peptide bond degradation, because then it will cleave any at a particular any place it wants. Okay, we won't get the selective or the selective degradation of the proteins if the enzyme has a broad specificity. We need the enzyme to be specific enough to cut on the particular bonds where we want. Okay. So let us discuss now little bit about the what is the proteases and what is the peptidases. Okay, so proteases or proteolysis, proteases or peptidases are enzymes that catalyze the breakdown of the peptide bonds in proteins or peptide. Proteolysis is the process of control or uncontrolled degradation of the proteins or peptides. Okay, so protease versus peptidase. Protease an enzyme which breaks down the protein and peptides. Peptidase an enzyme that breaks down peptides into amino acids. Okay, so basically, then this protease, an hydrolytic enzyme which hydrolyzes peptide bonds. Peptidase is a type of proteases. Okay, so peptidases are basically a part of proteases. Proteases are more broad, it has a more broad specificity, and this peptidases, these are what part of the proteases. Okay, so two types are proteases are there, endopeptidases and exopeptidases. And peptidase, if you I say only peptidase, it generally specifically means it is the exopeptidase. Okay, this proteases hydrolyze internal peptide bonds, and peptidases, it hydrolyze peptide bonds at the end. Proteases prefer intact protein. Okay, that's why statement number B is false, which says that proteases prefer small peptides. No, it is prefers the entire proteins. Peptidases, they prefer small peptides. This proteases, they result in oligopeptide, and peptidases result in a single amino acid or dipeptide. For example, pepsin, trypsin, chymotrypsin, papain, elastase, these are the example of proteases, and aminopeptidase, carboxypeptidase A, these are the example of peptidase. So let me give you an example. If let's say we have a chain of 10 amino acids, the proteases basically they will cut in middle, or they can cut in 2 to 3 places in the particular chain. But peptidases, it will specifically cut the last amino acid of the chain, which will result in a particular single amino acid, or sometime it can generate a dipeptide also. Okay, so these are the differences of proteases and peptidases. Most of the cases these are used as a single meaning. That's why sometimes you can get confused regarding this. Okay, I hope it is clear what is peptidase and what is protease. If you have any problem, any doubt, please kindly let me know in the chat box. Okay. So this was the problem number 1. Okay, you have tried to answer very good. Now let us go to the next problem. Okay. So what is also, let us discuss a little bit about these things also. So what is protease, peptidase, or proteinase? So protease is synonym with the peptidase, with almost the same, a little bit different. Peptidases are comprised of two group of enzyme, endopeptidases, exopeptidases, proteinases, which is, so basically this peptidase or protease, sometimes they are used in a similar terms. Okay. Now let us go to the next problem. Now problem number 2 says identify the correct structure for Sanger's reagents and Edman reagent. Your options are so this is number 1, this is number 2, this is number 3, this is number 4, and this is number 5. Now you have to identify the correct structures of the Sanger's reagent and Edman's reagent. Your options are number A is 1 and 2, number B is the 3 and 5, number C is 1 and 5, number D is 4 and 2. So identify the correct structures for the Sanger's reagents and Edman's reagent. Number 1 is this structure, number 2 is this structure, number 3 is this structure, number 4 is this one, number 5 is this one. So which one is Sanger's reagent and which one is Edman's reagent? Your options are number A 1 and 2, number B 3 and 5, number C 1 and 5, and number 4 D 4 and 2. I know all of you can answer this question because it's very easy. I will wait for a minute, then I will check the chat box. Identify the correct structures for Sanger's reagent and Edman's reagent. I want all of you to try to answer the questions. Even if your answer is wrong, it does not matter, but please try. So let me check the chat box. Okay, I have got two answer. One is saying it is number C, other is saying it is number A. Okay, let me check which of the following is the correct answer. So let me just make it full screen. Yeah. So the correct structure or the correct structures for the Sanger's reagent and Edman reagents are, the correct answer is number C, 1 and 5. So 1, this is the Sanger's reagent and this is the Edman's reagent. Okay, so number C was the correct answer. So this is Sanger's reagent, which is 1 fluoro 2,4-dinitrobenzene. Okay, this is 1, 2, 3, 4, 5, 6. If we go the numbering like this, so 1 fluoro 2,4-dinitrobenzene is known as the Sanger's reagent. Sanger's reagent, also known as 1 fluoro 2,4-dinitrobenzene or DNFB, is a chemical compound primarily used to identify N-terminal amino terminal amino acids of the polypeptide chain. It works by reacting with the free amino group of the N-terminal amino acid, forming a stable dinitrophenyl or DNP derivative. This derivative can then be separated and identified after the peptide chain is broken down by acid hydrolysis. Okay, this is Edman's reagent, or this is the phenyl isothiocyanate. Okay, phenyl isothiocyanate or PITC, the primary reagent used in the Edman degradation. So the primary reagent used in Edman degradation for protein sequences is phenyl isocyanate or PITC. It reacts with the N-terminal amino acids of the peptide or protein under the alkaline conditions to form a phenyl thiocarbonyl derivative or PTC derivative. This derivative is then cleaved under the acidic conditions, releasing the N-terminal amino acid as a phenyl thiohydantoin derivative or PTH derivative. Okay, so this is the Sanger's and Edman's reagent. We will discuss in detail how this Sanger's reagent and Edman reagents they work actually, and what does they do. We will discuss related regarding these things in today's class itself later. Okay, so the correct answer was number C. Okay, so just remember the structures of the Sanger's and Edman's reagent. Okay, now let us go to the next problem. Problem number 3 says which of the following statements regarding pepsin is false. Your options are number A, it was discovered in 1876 by William Kuhn. Number B, its acting site is the duodenum. Number C, it cleaves the peptide bonds at the C terminal end. And number D, it is an exopeptidase. Which of the following statements regarding pepsin is false? Number A, it was discovered by 1876 by William Kuhn. Number B, its acting site is duodenum. Number C, it cleaves the peptide bonds at the C terminal end. And number D, it is an exopeptidase. Which of the following statements regarding pepsin is false? Let me know. A, B, C or D, which of the following statements regarding pepsin you think is the false one? I will again wait for a minute, then I will check the chat box. As I have said, please try to answer the questions. Even if your answer is wrong, it does not matter. Which of the following statements regarding pepsin is false? Let me check the chat box. So let me check if there is any answer regarding your problem number 3. Okay, I have got few answers. Number C, number A, and okay, I have got two number A and one number C. Okay, let me check which of the following is correct answer. Okay, I think I have only got number A as the answer. So let me check. So the statement which is related to pepsin and which is false is it is number D, which says it is an exopeptidase. So basically, pepsin is an endopeptidase that breaks down proteins into smaller peptides. That is, it works as a protease. It is produced in the stomach and is one of the main digestive enzymes in the digestive system of humans and many animals. It helps the digest the proteins in the food. Okay, so basically, this proteolytic enzymes are of two type: endopeptidase and exopeptidase. Endopeptidase act in the interior of the protein. Exopeptidase act on the periphery of the protein. It cleaves the internal peptide bonds, and exopeptidase cleaves the terminal peptide bond. The end product of the endopeptidase is the small peptides. End product of the exopeptidase are free amino acids and dipeptides. Pepsin, renin, trypsin, chymotrypsin, elastase, these are the endopeptidase. Carboxypeptidase, aminopeptidase, these are the exopeptidase. So the statement number 1, which says it was discovered in 1876 by William Kuhn, it is correct. Its active site is duodenum. Although it is produced in stomach, it works on the duodenum. Okay, it cleaves the peptide bonds in the C terminal end. This is also true, okay, but it's clip the bonds on the C terminal end, but it cuts the chain in the middle, but in the C terminal end. And it is an exopeptidase. It is false because it is an endopeptidase. Okay, so the statement which is false is number D. Now let me go to the next problem. So problem number 4 says cyanogen bromide is specific in breaking the peptide bond associated with dash. Fill in the blanks with the correct amino acid. Your options are number A arginine, number B methionine, number C histidine, and number D proline. Cyanogen bromide is specific in breaking the peptide bond associated with dash. Fill in the blanks with the correct amino acid. Your options are number A arginine, number B methionine, number C histidine, and number D proline. So cyanogen bromide is specific for which amino acid cleavage? Arginine, methionine, histidine, or proline? Let me know in the chat box A, B, C, or D which of the following you think is the correct answer for problem number 4. Cyanogen bromide is specific in breaking the peptide bond associated with dash. Fill in the blanks with the correct amino acid. Your options are number A arginine, number B methionine, number C histidine, and number D proline. Let me check the chat box. Okay, so now let me check. Okay, I got one answer which is saying that it is number B. Okay. Okay. Let me now check what is the correct answer for the this problem number 4. I have got one answer which says it is number B. So now let me check which with the following is the correct answer. So the cyanogen bromide is specific in breaking the peptide bond associated with, very good, it is number B, methionine. Okay. So this is the structure of methionine. In our previous class, we have discussed methionine is basically a sulfur containing amino acid which has a SME bond at the terminal. So this is the cyanogen bromide cleavage. Okay, so let's say we have a particular amino acid polypeptide chain. Okay, this is the first amino acid, and this is the methionine. This is the second and third amino acid. Okay, so this is a basically example of tripeptide. Now cyanogen bromide, the first step, what it will do, this sulfur of this methionine, it will attack to the carbon of this particular CN. Okay, the CN bond carbon, it will attack, and the bromine, it will be leaving, and we will have something like this. Okay, so cyanogen bromide is basically the BRCN. Okay, the BRCN, it is known as a cyanogen bromide. And then we have this kind of intermediate which has this positive charge on sulfur with a methyl, and this is a CN here, and bromide is there in periphery. Then what will happen? The next amino acid which is on the right side of the methionine, means which is at the C terminal end of the methionine, what it will do, this nitrogen, it will just, it is written in this formation. What it will do, it will give the lone pair here, and this oxygen, it will then have this negative charge which will attack on this position, and this MESCN, it will just leave on from this thing, and we will have this 5-member kind of cyclic intermediate which has oxygen here, and this has the N positive, and Br negative is there as a counter anion. Okay, and then we have this water. If we do the water hydrolysis, what we will have this C terminal peptide fragment, it will just go, and we have this homoserine lactone unit which is a N terminal peptide fragment. Okay, so that's how cyanogen bromide fragment create the fragments from a polypeptide chain wherever there is a methionine. Okay, it cuts on the C terminal end, and we get this kind of homoserine lacting unit of the N terminal end and C terminal peptide fragment. So this is example of the non-enzymatic cleavage. Okay, so non-enzymatic cleavage of methionine residue by cyanogen bromide in presence of cysteine. Also, it can, so although nucleophilic sulfur of methionine is responsible for attacking the cyanogen bromide, the sulfur of cysteine does not behave similarly. So this is the important thing. Although methionine and cysteine both have sulfur, but the sulfur of cysteine does not behave similarly that of the of the methionine. If the sulfur in cysteine attacks cyanogen bromide, the bromide ion will deprotonate the cyanide adapt, leaving the sulfur uncharged, and the beta carbon of the cysteine does not electrophile. So the strongest electrophile would let them the cyanide nitrogen that will attack, if attacked by water, will yield a cyanic acid and the original cysteine. Okay, so if it happens with cysteine, this is an example. So what will happen if there is a water? We will get the original peptide or original cysteine, and we also and the cyanogen bromide, it will convert it to the cyanic acid. So this is the mechanism how it happens in case of the cysteine. Okay, so in case of methionine it happens like this, in case of cysteine it happens like this, the cyanogen bromide. So this is very important thing, so remember, and you have to practice the mechanism 1 or 2 times to get it correct. Okay, so I think this was an problem number 4. So if there is any doubt, please let me know. If you do not have understood the mechanism or anything, please kindly let me know. I will repeat the thing. If not, then I will proceed with the next question. Okay. So now let me go to the next question. Problem number 5, it says that which of the following statements regarding Sanger's method is false? We have discussed and I have told you what is Sanger's reagent. Now this question is related to the Sanger's method. So let me know which of the following statements regarding Sanger's method is false. Your options are: number A, this method is used to determine the amino acid from the N-terminal end. Number B, this reagent used in Sanger's method is 2,4-dinitrofluorobenzene. Number C, the Sanger's method cannot easily distinguish between a cyclic and an acyclic peptide. And number D, the amino acids are identified with the help of HPLC in the end of this method. Which of the following statements regarding Sanger's method is false? Read the options very carefully and let me know in the chat box A, B, C or D which of the following you think is the correct answer. Number 8, this method is used to determine the amino acid from the N-terminal end. Number B, the reagent used in Sanger's method is 2,4-dinitrofluorobenzene. Number C, Sanger's method cannot easily distinguish between a cyclic and acyclic peptide. Number D, the amino acids are identified with the help of HPLC at the end of the method. I will wait for some time, then I will show you the answer and we will discuss about the Sanger's method. So which of the following statements regarding Sanger's method is false? Let me check the chat box if there is any answer. Let me check. Okay, another answer I have got for this problem number 5 is now, which is saying it's number C. Others are not answering. Only I find one person answering the questions. Other also should answer the questions. Okay. So now let me check which of the following will be the correct answer for our problem number 5. So the correct answer for problem number 5 means the statement regarding Sanger's method is false. The statement is very good. It is number C. Sanger's method cannot easily distinguish between a cyclic and an acyclic peptide. This statement is false. Why it is false? Now let us discuss. So what is Sanger's method? So Friedrich Sanger had demonstrated a method to determine the amino acid residue located on the N-terminal end of the polypeptide chain by using the reagent called fluoro-dinitrobenzene FNB or 1-fluoro-2,4-dinitrobenzene also you can say, or the 2,4-dinitrofluorobenzene. Everything is correct. Okay. So Sanger showed for the first time that amino acids are covalently together by alpha amino and alpha carboxyl group. He suggested stepwise release and identification of amino acid starting from the N-terminal. He used a reagent called fluorodinitrobenzene or dinitrofluorobenzene, which is commonly called as the known as the Sanger's reagent. This reagent reacts with the free NH2 group of the N-terminal end. Upon hydrolysis, yellow colored DNP derivative of the amino acid is produced. So this is the 2,4-dinitrofluorobenzene or you can say fluorodinitrobenzene. Okay, so this is the Sanger's reagent. This is a polypeptide chain. So at the end terminal of the polypeptide chain, we know there is a free amine group. So what does this do? This amine nitrogen, it attacks on this position and the fluorine leaves as a leaving group, and we have this particular DNA polypeptide, DNP polypeptide. Upon hydrolysis, this particular bond is cleaved, okay, and this will be separated, and this particular first amino acid will be now then separated, and it can be identified using the HPLC method. Okay, so if there is a cyclic peptide, let us say there is a cyclic peptide, so that particular cyclic peptide does not have any free NH2 or free carboxylic acid or free N terminal or C terminal end. That means it does not have any free NH2 group. If there is no free NH2 group in the there is no free NH2 group at the N terminal end, then this Sanger's reagent cannot react with that particular peptide. So that's how we can distinguish between a cyclic and acyclic peptide by using the Sanger's method. Okay, that is why the statement number C, which says that Sanger's method cannot easily distinguish between a cyclic and acyclic polypeptide, this is not correct. This is false. Okay. So there is any doubt, any query? I hope okay, no, there is no problem. So now let me go to the next problem. If there is any no doubt in this particular problem number 5. So problem number 6, it says that which of the following derivative is formed in the Edman degradation of the polypeptide chain. We have discussed, I have shown you what is the Edman's reagent. Now you have to identify what derivative it will form the Edman degradation. Your options are number A DNP derivative, number B PTH derivative, number C DCC derivative, and number D DNS derivative. Which of the following derivative is formed in the Edman degradation of the polypeptide chain? DNP derivative, PTH derivative, DCC derivative, or DNS derivative? I will wait for a minute, then I will check the chat box. Try to answer the question. Which of the following derivative is formed in the Edman degradation of polypeptide chain? DNP derivative, PTH derivative, DCC derivative, or DNS derivative? So, which of the following derivative is formed in the Edman degradation of polypeptide chain? Let me check if there is any answer in the chat box. I have got one answer which says it is number B. Okay, let me check if number B is the correct answer or not. Again, I am saying please try to answer the questions. Okay. So, the derivative which is formed in the Edman degradation of polypeptide chain will be, yes, it is number B, PTH derivative. Okay. So, now let me show you how this Edman degradation happens. Basically, so this is, so first let us understand the structure. So, this is known as the hydantoin. This is known as the thiohydantoin. Why I have shown you the structure? I will just tell you in a minute. So this is the Edman degradation of the polypeptide chain. Okay, so this is our Edman's reagent, the which is known as the phenyl thioisocyanate. Okay, then this is our polypeptide chain. This PTH derivative is formed, or this Edman reagent reacts to the same mechanism or the same thing like as the Sanger's reagent. Okay, there is a N-terminal free NH2 group. This NH2 group attacks on this carbon, and then this kind of electron shift happens. Okay, and then we have this lone pair on nitrogen, and this nitrogen lone pair again gives this electron on this bond in carbon nitrogen bond. Okay, and then what happens, this double bond of sulfur, this goes to the next C carbonyl group of the amino carbonyl group of this peptide chain, and this get hydrolyzed, and after water hydrolysis we have this kind of derivative which is formed with the N-terminal amino acid and the Edman's reagent, and this is known as the phenyl thiohydantoin derivative. Okay, this is known as the phenyl thiohydantoin derivative. So this is a hydantoin which has two oxygen, two nitrogens. Thiohydantoin is have one thio in place of the oxygen, and this is also the phenyl thiohydantoin derivative. Okay, and this R group basically is based on whatever this the N-terminal amino acid. For example, if alanine is the N-terminal amino acid, then R will be methyl, right? So that's how this Edman's reagent also identify what is the N-terminal amino acid, just like our Sanger's reagent do. Okay, so this is how it works. So this derivative is known as the PTH derivative, whereas the Sanger's reagent, it is the N-terminal amino acid forms the DNP derivative. The name, so you have to remember what is the method. Sanger's method, Edman's method, what is the method, what is the reagent, what is the name, what is the structure, and what is the mechanism, and what does it basically do. So these things you have to remember in detail. Okay, the mechanism, it may look sometime it is complicated, but if you practice it one or two times in your copy, you will find it is very easy mechanism. There is nothing to worry about. Okay. So is there any doubt or is there any query? I find only one student is answering all the questions. I want all of you to participate and answer. Okay. Even if your answer is wrong, it does not matter. I have already told you that I am not going to give you any mark if your or negative marking if your answer is wrong. But please try to answer during the class. Okay. Let us move to the next question. Problem number 7. Problem number 7 says, which of the following statements is false regarding pepsin and trypsin? Your options are: number A, trypsin is produced by the gastric glands while pepsin is produced by the pancreas. Number B, trypsin is secreted into the duodenum of the small intestine. Number C, pepsin functions in an acidic medium. Number D, there are two types of trypsin. Which of the following statements is false regarding pepsin and trypsin? Again, I will repeat. Number A, trypsin is produced by the gastric glands while pepsin is produced by the pancreas. Number B, trypsin is secreted into the duodenum of the small intestine. Number C, pepsin functions in the acidic medium. Number D, there are two types of trypsin. Which of the following statements regarding pepsin and trypsin is false? Which of the following statements is false regarding pepsin and trypsin? I will wait for another minute, then I will check the chat box. Let me check if there are any answer for problem number 7. Okay, I got two answers. One is saying it is number A, another is saying it is number D. Let me check which of the following is the correct answer, A or D. So, the statement which is false regarding pepsin and trypsin is number A. Very good. There was this answer in the chat box, I think. So, trypsin is produced by gastric glands, pepsin is produced by pancreas. This statement is false. Now let us discuss about pepsin and trypsin. So basically pepsin, it is the chief digestive enzyme in the stomach which breaks down the proteins into polypeptides, and trypsin digestive enzyme which breaks down proteins in the small intestine secreted by the pancreas as the trypsinogen. Okay, this is produced in the gastric glands, the pepsin, and the trypsin is produced by the pancreas. So that is why the statement number A is false, which is saying that trypsin is produced by gastric glands and pepsin is produced by pancreas. It is not, it is the other way. Trypsin is produced by the pancreas and pepsin is produced by the gastric glands. Okay. So the component, pepsin is a component of the gastric juice, trypsin is a component of the pancreatic juice. Pepsin is secreted into the stomach, trypsin is secreted into the duodenum of the small intestine. Pepsin is inactive form is pepsinogen and trypsin's inactive form is trypsinogen. Inactive form of the pepsin is activated by HCl of the gastric juice. Inactive form of the trypsin is activated by an enzyme called the enteropeptidase large enzymes. Okay. The pepsin, four types of pepsins are there, pepsin A, B, C and D, and two types of trypsins are there, alpha and beta trypsin. Pepsins functions in the acidic medium, trypsins functions in the alkali medium. This is very important to remember. Pepsin hydrolyzes peptide bonds between the large hydrophobic amino acid residues and trypsin hydrolyzes the peptide bonds at the C-terminal side of the lysine or arginine. Pepsin is responsible for the converting proteins into proteases or peptones, and trypsin is responsible for the converting proteins into polypeptides. Pepsin is used in digestion of the antibodies, preparation of the collagen, protein digestibility assays, and subculturing viable mammillary epithelial cells. Whereas trypsin used in tissue dissociation, cell harvesting, mitochondrial isolation, in vitro protein studies. Okay. So these are the differences of the pepsin and trypsin. So the few points which you need to remember. If you go through this table, I think it more or less summarizes all the related things related to pepsin and trypsin. So is there any doubt? If there is any doubt, please let me know. Okay, I will answer the doubts at the end of the session if there is any, or in between also we can discuss that particular doubt. Now let us go to the next problem. Problem number, okay, so there was a mistake in numbering. Last one, I think it was our problem number, okay, this one the problem number 7 actually. And this was the problem number 7. Now we will going to discuss the problem number 8. There was an issue in numbering. I will correct it later. So this question, it says that which of the following amino acids restricts the action of chymotrypsin? Your options are number A alanine, number B serine, number C proline and number D methionine. Which of the following amino acid restricts the action of chymotrypsin? Alanine, serine, proline or methionine? Let me know in the chat box. A, B, C or D, which of the following is the correct answer? Which of the following amino acids restricts the action of chymotrypsin? Alanine, serine, proline, methionine, which will be the correct answer? Which of the following amino acids restricts the action of chymotrypsin? Alanine, serine, proline or methionine? Please try to answer the questions. As I said, it does not matter if it is right, is your answer wrong or right. Right. Which of the following amino acids restricts the action of chymotrypsin? Alanine, serine, proline or methionine? Let me check if there is any answer for that particular question. Okay, so this is the problem number 7, I think. Yeah. Okay, I got two answer. One is saying it is number C, other is saying it is number D. Okay, now let me check which of the following will be the correct answer, C or D, or if there is any other correct answer. So the correct answer, the amino acid that restricts the action of chymotrypsin is, that is proline. Okay, so yeah, there was a correct answer in the chat box. So proline is the amino acid which restricts the action of the chymotrypsin. So this is the polypeptide chain, and this is the action of the chymotrypsin. So what does the particular chymotrypsin do? So chymotrypsin breaks the polypeptide chain or the peptide bond involving the aromatic amino acid at the carboxylic end. About chymotrypsin, one thing you have to remember is that the adjacent amino acid has one restriction. If the amino acid happens to be next to proline, then hydrolysis cannot take place. So the next amino acid that is connected to the aromatic amino acid if it happens to be proline, the hydrolysis cannot take place. Let's say this is the example. So let us say this is the R. This is the first amino acid is this one, and the second amino acid is this one. Let's say, okay. What does this particular this polypeptide chain, how it will the cleave at the C terminal end? Okay, the C terminal end it will cleave, and this position. Let me just use the pen. So chymotrypsin, what it will do, it will cleave at the C terminal end if R is the aromatic amino acid like phenyl, tryptophan and tyrosine, but provided R dash is not proline. If R dash is proline, then this particular cleavage cannot take place. Okay, so this thing you have to remember. So this particular question has two particular important things, that chymotrypsin that binds in the peptide bond which involves aromatic amino acid at the so it binds, it cleaves the C terminal end of the aromatic amino acid, but the next amino acid cannot be proline. Otherwise, this hydrolysis cannot take place. Okay. So this thing you have to remember. So this was the problem number 8. Now let me go to the next problem. Problem number 9 says, which of the following protecting group is not used to protect the amine group? Your options are number A, BOC, BOC, number B, TBDMS, number C, FMOC, and number D, CBZ. Which of the following protecting group is not used to protect the amine functional group? BOC, TBDMS, FMOC, or CBZ? So basically, this now we are going to move into our protection reprotection chemistry, which is a very important part of the organic chemistry or also of the protein chemistry. The protection reprotection. Why it is important? We will come shortly to that point. First, let us discuss a little bit about the different protecting groups. So the first, problem number 9 says, which of the following protecting group is not used to protect the amine functional group? Your options are number A, BOC, number B, TBDMS, number C, FMOC, number D, CBZ. Let me know. I will wait for some time. Which of the following protecting group is not used to protect the amine functional group? BOC, TBDMS, FMOC, or CBZ? Let me check the chat box. So is there any answer? Okay. So for the question, we don't have any answer. So now let me show you the answer and let us discuss about it. So the protecting group which is not used to protect the amine functional group is, it is number B, TBDMS. So basically TBDMS, it basically protects the alcohol functional group. Okay. So basically this is the example. So this is the TBDMS, which is the tertiary butyl dimethyl silyl chloride. Okay, so this is the DMSCL. Okay, also it is known as, this is DMSCL. This is a short form. So this is the structure of this TBDMSCL, which is the silicon, which is tertiary butyl 2 methyl groups. Okay, that is known as the tertiary butyl dimethyl silyl chloride. So this is the alcohol functional group. What does it do? Basically, it protects the alcohol functional group, which is known as the TBDMS protection. This is how this particular protection happens. Because silicon has a strong affinity to the electronegative element, particularly oxygen, fluorine, and chlorine. So trialkyl silyl ethers are attacked by the hydroxyl ion or water, not fluoride ion, but are more stable to carbon and nitrogen bases and nucleophile. They are usually removed by the aqueous acid or fluoride salt, particularly TBUF or tetra N-butyl ammonium fluoride. Okay, so which is soluble in the organic solvents. In fact, TBDMS is one of the member of the whole family of the trialkyl silyl protecting groups, and the relative stability of the nucleophiles of the various kinds is determined by the three alkyl groups carried by silicon. The most labile TMS trimethyl silyl is moved simply on treatment of methanol, which is the most stable required hydrofluoric acid. So this is the how this deprotection of this happen. This is the this is the TBDMS protected alcohol group, and then you can use the TBUF. TBUF will attack the fluoride, will attack the silicon, and we will have the deprotected alcohol. Also, you can use the acid, which also can use this particular deprotection. Okay, so the protecting group TBDMS structure is this. The protects alcohol from nucleophiles of C nitrogen bases, carbon or nitrogen bases. Protection requires this, and deprotection requires these things. Okay, so this is how this protection deprotection works. So this is there is a table. So this is a very important table which you can remember. So it will help you for remembering different protection and deprotection groups. Like for ketone and aldehydes, acetal or dioxylones can be used. Okay, so this this group, acetal group, is used to protect the ketones from nucleophiles or bases. To protect, we require this, and to deprotect, we require proton and H2O. Then TBDMS I have discussed already. What does it do? We have THP. THP protection group is also used to protect the alcohols from strong bases, and you require the dihydropyran and acid. And for the protection, you require H plus and H2O. Benzyl ether. It also protects the alcohol from almost everything. This is a very good protection group. And for to protect, you need the benzyl bromide and sodium hydride. To deprotect, you need the hydrogen, palladium, carbon, and HBr. Okay. Then we have this methyl ether also, which is similar to our benzyl ether. And then we have this tertiary butyl ester, which protects this tertiary butyl ester protects the carboxylic acid groups from nucleophile. To protect, you need the isobutane or H plus, and to deprotect, you need the strong acid. Then we have CBZ protecting group, which protects amine. CBZ, BOC protect TBOC and FMOC. All these things are protects the amines from the electrophiles. For CBZ protection, you need the BNOCOCl and base. To deprotect, you require HBr in acetic acid or H2 palladium. For TBOC, so BOC protection, you need this BOC anhydride and base, and to deprotect, you need the H plus H2O, and FMOC, you need the FMOCCl and base and amine. So these are actually this table is very good because it summarizes all the protecting and deprotecting groups which you need to remember for the answering this kind of protection deprotection questions. Okay, so this was the problem number 9. So there is any doubt? Let me check the chat box once. I hope there is no doubt. If there is any doubt, please let me know. This is a very easy things, but you just need to remember because there are too many things. Sometimes it may seem that there is a lot, but if you read and you will find that there is not much. That you need to just remember these. What to remember? I mean, what to protect from, what you need to protect, and what to need to deprotect. Okay. Let us go to the next problem. Problem number 10 says, which of the following statements regarding the FMOC protection is false? Your options are: number A, FMOC stands for the 9-fluorine methoxycarbonyl chloride. Number B, in case of the deprotection, aqueous piperidine is used. Number C, inorganic bases can also be used to the protection of FMOC protected amines. Number D, the extent of the deprotection can be quantified with the help of the HPLC in this method. Which of the following statements regarding FMOC deprotection is false? Your options are number A, FMOC stand for the 9-fluoronyl methoxycarbonyl chloride. Number B, in case of deprotection, aqueous piperidine is used. Number C, inorganic bases can also be used for deprotection of the FMOC protected amines. And number D, the extent of the deprotection can be quantified with the help of the HPLC in this method. Which of the following statements regarding FMOC deprotection is false? Let me know in the chat box A, B, C or D which of the following you think is the correct answer. Let me check the chat box. Okay, there is no answer. Okay, let me check. Let me show you the correct answer and let us discuss about it. So, the statement which is false regarding the FMOC protection is number C, inorganic bases can also be used for the protection of FMOC protected amines. This statement is false. Okay, so FMOC basically it stands for the fluorine methoxycarbonyl chloride or FMOCCl. This is the structure of FMOCCl. It is a very good reagent and very widely used in case of the protection of the protection of the free amine group and basically of the amino acid. This is a very widely used. Mostly it is used FMOCCl in case of our peptide chemistry. Okay, so just remember the structure. What is the full structure? What is the full form? This is very required. So FMOC protection group. So this has a susceptibility inverse to that of the BOC. FMOC or fluorine methynyl carboxycarbonate protecting group cannot be lost by the substitution in the manner of CBZ or TBOC because neither S1 or S2 SN2 mechanism can operate at the rigid carbon both primary and hindered. Okay. So, neither this carbon we are talking about. So this is a FMOC protected amine, right? Or FMOC protected amine. So neither SN1 or SN2 can take place in this carbon because the compost. That is why this particular FMOC protected amines are stable to acid. So where is the safety catch? The FMOX Achilles heel is rather the acidic proton, which is this one. Now about pK of 25. Okay, this acidic because the anionic product is aromatic product of deprotection is aromatic. Only a very small concentration of aromatic anion ever forms, but it does not immediately undergoes elimination. FMOC protected amines are readily deprotected in base. Okay, so you have to add a little base, and mostly organic bases are required, organic nitrogenous bases, which can deprotonate this position and this anion. Then particular fluorine this group is eliminated and we have this deprotected amine. After it goes to the it releases the carbon dioxide. Okay, and also one thing, this particular group is can be quantified with the help of HPLC. Okay, so this is the all about the FMOC protection and deprotection. This is a very important. Okay, the FMOC protection, FMOC deprotection, and the deprotection is very important. You have to remember what is the FMOCCl, what is the structure, what is the mechanism, and that's these things. Okay. Again, I am saying this mechanisms, it may seem sometime that it is a little complicated, but if you practice it 1 or 2 times, then you will find that this is very easy. Okay, this is there is no complication, or this is not nothing to worry about. But you have to practice it 2 to 4 times. So let us go to the next problem. Problem number 11 says, which of the following is used to deprotect tertiary butyl ester to avail the free carboxylic acid group? Your options are number A strong acid, number B strong base, number C inorganic salt, number D hydrogen palladium. Which of the following is used to deprotect the tertiary butyl ester to avail the free carboxylic acid group? Strong acid, strong base, inorganic salts, or hydrogen palladium? Which of the following is used to deprotect the tertiary butyl ester to avail the free carboxylic acid group? Strong acid, strong base, inorganic salts, or hydrogen palladium? Which of the following is used to deprotect the tertiary butyl ester to avail the free carboxylic acid group? Okay, let me check the chat box, or should I wait for 1 minute? Let me check if there is any answer. Okay, I got one answer which is saying it is number A. Let me check if number A is the correct answer. So, to deprotect the tertiary butyl ester and to avail the free carboxylic acid group, we require number A, strong acid. So the more commonly used carboxylic acid protecting group is rather more stable toward the acid attack by nucleophile, that is the tertiary butyl ester. Tertiary butyl ester can be made by reacting the carboxylic acid with the carbon generated from the isobutene in sulfuric acid. So this is the isobutene in sulfuric acid. We will have this tertiary butyl ester of the carboxylic acid, and this is a very good protecting group for the thing, and this is how it happens. This is the mechanism. So first this double bond, it takes up a proton of the in strong acid high sulfuric acid medium, and then this proton is the carbocation is formed here, and then we have this carboxylic acid group and reacts like this, and we get this ester. So steric bulk of this particular tertiary butyl group, okay, means this actually means that the tertiary butyl esters are resistant to nucleophilic attack on the carbonyl group. Okay, so this is the particular bulk of this tertiary butyl group makes the nucleophiles, they actually restricts the nucleophile to attack this particular carbonyl carbon group, okay, including the hydrolysis under the basic condition, means the nucleophilic attack by hydroxyl anion. But they do hydrolyze relatively easily in the acid because the mechanism of hydrolysis of tertiary butyl esters in acid is quite different. Instead, undergoing nucleophilic attack, this carbonyl group, this tertiary butyl ester loses a stable carbocation, and which is either captured by the solvent in an SN1 reaction or loses the proton in an E2-1 reaction. So if there is a strong acid medium, we will have this proton. This is the protonation of this carbonyl carbon group, and then what happened? This group, this bond actually breaks to make a very stable carbocation, this tertiary butyl carbocation. It can either participate in the E1 elimination reaction, which will give us the isobutene back, or it can participate with the reaction, this SN1 reaction with the solvent, which gives us this one. Okay, so you have to remember tertiary butyl ester is the protection group. It protects, the structure is this. Okay, it protects the carboxylic acids from nucleophile, and to protect you need the isobutene and in a acidic medium. To deprotect you need strong acid. Okay, so this one all about the protection of the carboxylic acid groups, which uses this tertiary butyl ester. Again, you have to practice the mechanism one or two times to get it perfect. Okay. Now let us go to the next problem. Problem number 12 says, which of the following reagents are suitable for the deprotonation of the following? So this is the thing which you need to deprotect. Okay, so this is the CBZ protecting group. So as you can see, this is the leucine. We have this benzoyl chloroformate or BNO-COCl, and this is the base which gives us the CBZ protecting group. Okay, so which of the following reagent is suitable for the deprotection of the following? Your options are number A hydrogen palladium, number B HBr ACOH, number C none, and number D both. Which of the following are suitable for the protection of this group? Which of the following reagents are suitable for the protection of the following? Hydrogen palladium, HBr ACOH, none or both? Let me know in the chat box ABC which of the following you think is the correct answer. Which of the following reagents are suitable for the protection of following? Let me know. Let me check the chat box. Okay, I got one answer again, which says that it is number A. Let me check if number A is the correct answer or not. So the reagent which is used for the protection is, correct answer is number D. Both hydrogen palladium and HBr acetic acid can be used to protect the CBZ protection group. So this is the CBZ. Just let me show you. Yeah. So CBZ is basically this group. Okay, this is this OCO, OCO or okay. So this is a benzyl group, and then this is the OCO. So this is the CBZ protection group. This is the structure of the CBZ protected amine. So it protects amine from electrophiles, and the protection you read this benzyl chloroformate and base. And to deprotect you need hydrogen acid, HBr acetic acid you can use, also you can use hydrogen palladium. The group that was used, one of the most important nitrogen protecting group, and is known as the CBZ protecting group. CBZ stands for the carboxybenzyl. Now CBZ groups are put by treating the benzyl chloroformate and the weak base. CBZ protection amines are actually carbamates, just like amide. They are no longer nucleophilic because nitrogen's lone pair is tied in a conjugation with the carbonyl group. They are resistant to both aqueous acid and aqueous base, but they have used analogy we have developed earlier is an acyl, that is the benzyl group. This benzyl group is the acyl for the I mean the weakest weak point for this particular group. So what is the mechanism of deprotection? So cleavage of the CBZ by HBr acetic acid. So what does it happen? How it happens? So HBr is a strong acid. Okay, so it have this proton. This carbonyl group is protonated at first. Then what happens? This bromine, it attacks on this benzyl position. This Br minus, which is a good nucleophile, it attacks on the benzyl position. We have the this kind of mechanism which gives us this thing, and then we can what we can have is with this after taking up this proton, there is a elimination of carbon dioxide, and we have this internal proton abstraction makes this anion, and this anion loses this then this kind of carbon CO2 elimination happens, and we have this free amine. By hydrogenolysis, what how does it happen? Using hydrogen palladium, what does it do? There is a benzylic CO bond which is cleaved, okay, by via hydrogenolysis, and then we have this toluene, okay, and this and then we have this again. Similarly, we have an internal proton abstraction, elimination of carbon dioxide, and free NH2 group. So that's how this deprotection of this particular CBZ protecting group happens in case of using both HBr acetic acid and also using hydrogen palladium. Okay, so this was how the deprotection happens. Now let me go to the next problem. Problem number 13 says, which of the following reagents can convert the carboxylic acid directly to the alcohol? Your options are number A, NaBH4, number B, Br3, number C, sodium CnBH3, sodium cyanoborohydrate, and then we have this lithium borohydrate. Which of the following reagent can convert the carboxylic acid groups directly to the alcohol group? Sodium borohydrate, Br3, sodium cyanoborohydrate, lithium borohydrate. Which of the following reagents can convert carboxylic acid directly to alcohol? Let me know. Which of the following reagent can convert carboxylic acid directly to the alcohol? Sodium borohydrate, Br3, sodium cyanoborohydrate, lithium borohydrate. Let me wait for a minute, then I will check the chat box. Which of the following reagents can you be used to convert the carboxylic acid to alcohol directly? Let me check the chat box. Let me see if there is any answer. An answer I got which is saying it is number D. Next, let me check if number D is the correct answer or not. So the reagent which is used to convert carboxylic acid directly to alcohol is number B. It is Br3. So how to reduce carboxylic acids to alcohol? So the best reagent is the borane or BH3. Borane is in fact a gas without the with the structure of B2H6, but it can be tamed as a liquid by complexing with ether. Okay. or THF or dimethyl sulfide or DMS. Okay, although boron appears superficially similar to borohydrate, it is not charged, and that is it makes a difference with the reactivity. Whereas borohydrate reacts best with the most electrophilic carbonyl groups, boron's reactivity dominated by its desire to accept the electron pair into the boron's empty p orbital. Now, it is the context of the carbonyl group reductions. This means boron reacts reduces electron-rich carbonyl groups fastest. Carbonyl groups of acyl chloride and esters are relatively electron poor because of the chloride and OR are very electronegative. Boranes will not touch acyl chloride and reduces ester very slowly, but it will reduce very effectively carboxylic acids and amides. Boron reacts with the carboxylic acid first of all to form the triacyl borates with the evolution of hydrogen gas. So this is the triacyl borate is the structure and hydrogen gas is evolved. Then esters are usually less electrophilic than the ketones because the conjugation between carbonyl group and the lone pair of sp3 hybridized oxygen atom. But in this boron ester, the hydroxygen is next to the boron, which is share the lone pair in the carbonyl group and when boron's MTP orbital. So they have considerably more reactive than normal esters. Okay, so basically they have the boron has the MTP orbital and this carbonyl, this carboxylic acid. These groups are very electron rich. Okay, that is why it can easily react and the formed adducts are very stable. So carbonyl reduction using the hydride reducing agent, we have this imine, aldehyde, ketone, ester, amide and carboxylic acid. Imines are very the most, so the electrophilicity runs goes like this. This is the more electron deficient, means these are the more reactive. Okay, imine, acyl chloride, these are the more reactive, but amide carboxylic acid they are less reactive. Imines can be reduced by every one of them, and it forms this. Then we have this aldehyde. Aldehyde is reduced slowly with the sodium cyanoborohydrate, but a very fast reduction happens with sodium borohydrate, lithium aluminium hydrate, and then slow reduction with the this borons or boranes. Okay, then we have this particular ketone, which is the similar reactivity looks shows like aldehyde. Esters cannot be reduced by so ester, amide, carboxylic acid. This cannot be reduced by sodium cyanoborohydrate. Slowly, esters can be reduced by sodium borohydrate, but amide and carboxylic acid they cannot be reduced by these hydrides, which charged hydrides. Okay, but amide and carboxylic acids can be reduced easily using boranes. Okay, and esters can be converted to our aldehyde. Okay, via diol and by amides can be converted to our aldehyde using lithium aluminium hydrate. Carboxylic acids can be reduced to our carboxylic acid can be converted to the aldehyde via acyl chlorides to form the aldehyde. So these are the chemoselectivity of the reduction of carbonyl groups, and this is very important. Okay, this table and these things are very important to remember and to because this sometimes most of the students they get confused regarding because we have lots of reducing agent which to use, where to use. So these are the complete chart which you need to remember. Now I will go to the next problem. Problem number 14. It says which of the following is the most important thing that should be kept in mind for the peptide synthesis. Your options are number A, choice of amino acid, number B, choice of coupling reagent, number C, choice of protecting group, and number D, it varies sequence to sequence. Which of the following is most important thing that should be kept in mind for the peptide synthesis? Number A, choice of amino acid, number B, choice of coupling reagent, number C, choice of protection group or protecting group, and number D, it varies sequence to sequence. I will wait for a minute, then I will check the chat box. Let me know A, B, C or D, which of the following you think is the correct answer. Which of the following is the most important thing that should be kept in mind for peptide synthesis? What? Which of the following is the most important thing that should be kept in mind for peptide synthesis? Number A, choice of amino acids. Number B, choice of protect coupling reagent. Number C, choice of protecting group. And number D, it varies sequence to sequence. Let me check the chat box now. So, is there any answer for that last question? I think there is no answer for the previous question. Okay, let me show you what is the correct answer and let us discuss. So, the most important thing that should be kept in mind for peptide synthesis is number C, the choice of protecting group. So, peptide synthesis has become one of the most reliable and predictable fields of the practical organic chemistry, principally because of the effectiveness of the protecting groups it employs. For this reason, peptides are one of the few classes of complex organic molecules that can routinely be made by machines, such as one of the in which biology makes the peptides and proteins selectively coupling together members of the pool of 20 or so amino acids. To do the same in laboratory, we need to overcome number of challenges. For example, we will start by thinking how to react two amino acids together to make a dipeptide, leucine and glycine, for example. So this is the leucine and this is the glycine. Okay, if we want that NH2 group of glycine to be react with the carboxylic group of the leucine, we first need to activate the carboxylic acid toward the nucleophilic substitution by making acyl chloride or say to particularly react extra. So to make this dipeptide, first we have to make this particular carboxylic acid group we have to activate it to make it the acyl, but the acyl chloride are not used for peptide synthesis because the potential of the unwanted side reaction including the decimization or dimerization of the product by making the diketopiperazine. Piperazine. The main problem is that there is another free carboxylic group, this one, which will react with the CH2 group to make the anhydride and two different amines that the free might get giving the both to get our the homo product that the leucine or leucine glycine we require, but leucine can also be made by this particular for that reason we need to protect the NH2 group of leucine because we do not want this to react and to protect the COOH group of glycine and this one. So this and this two need to be protected first before coupling of these two. Okay, so what sort of protecting groups need to be used? We still need to be able to take them off again once we done their job. So this is an important thing that say amide to protect the amine, since we would have great difficulty hydrolyzing the amide in presence of the amide which are trying to form. Not only do we want a protecting group to be removable in under mild condition, we also want two groups, one of for the CH2, NH2 and COOH. We want to take off under different condition. Then we have the opportunity to modify either end of the dipeptide as our will. Okay, so we need to protect the amine of this, the protecting group using the protecting group 1 and also using the protecting group 2 we can protect the glycine carboxylic acid after the making of this particular peptide bond using the acid or if it is removable only in acid, then in acid we will have this further which we can use further for coupling, or then we can use this another acid to reprotect this portion. Okay, so to make it ready for coupling with the another part. So the good choice of a pair of condition might be acid or base. Okay, so this thing is very important. How you are protecting your amine group, how you are protecting your carboxylic acid group is very important in case of the peptide bond formation. The choice of protecting group is the most important thing that should be kept in mind for the protein synthesis. Okay, others may vary, others may change and everything, but the choice of the protection and deprotection groups are very important. Okay. Now I will go to the next problem. Problem number 15 says, which of the following is not a phosphorus-based coupling agent? Your options are number A, PIBOP, number B, DCC, number C, HBTU, and number D, HATU. Which of the following is not a phosphorus-based coupling agent? PIBOB, DCC, HBTU, or HATU? Again, I will wait for a minute. Try to answer the question. Which of the following is not a phosphorus-based coupling agent? PIBOB, DCC, HBTU, or HATU? Coupling agents are basically used to, for efficient coupling between two amino acids to form a peptide bond. Sometimes the groups, the carboxylic acid groups or the particular your amine groups, they need to be they need to be activated for the peptide bond formation. That's why coupling reagents are used. You have to identify which of the following is not a phosphorus-based coupling agent. I will go a little fast from now because we have 6 questions more, I think, including it, which we need to cover up within 30 minutes. Which of the following is not a phosphorus-based coupling agent? Try to answer the question in the chat box. A, B, C, or D. Which of the following you think is the correct answer? Let me know. Let me check if there is any answer given. Okay, I got one answer which is saying it is number B. Let me check if number B is the correct answer or not. So the correct answer will be yes, number B, DCC. So these are the structures for this coupling reagents. Okay. This is DCC, which is not a phosphorus-based coupling agent, whereas PIBOP, HBTU, HATU, all these are phosphorus-based coupling reagent. So simple dipeptide synthesis, peptides are amide linkage that connect amino acids. Amino acids have at least two reactive functional group, amine and carboxylic acid. Okay, in order to peptide coupling to proceed to high yield, appropriate protecting groups must be used with one carboxylic acid and other amine. Otherwise, we get the mixture of dipeptides from amino acid A and B. We will get A, B, B, and B. So these are the four different, possibilities which can generate from the two amino acid. Typically, one carboxylic acid is protected as an ester and one amine is protected as a carbamate. This allowed the desired coupling to be processed significantly. This amine is protected as a carbamate, carboxylic is protected as a ester. Using DCC coupling agent, we have this thing. Okay, so this is DCC. DCC full form is NN dicyclohexyl carbodiimide. Okay, this is, and it after the peptide formation, it forms the DHU. Okay, so this DHU is basically nothing but there is a hydrogen, there is oxygen and nitrogen here. Okay, the dicyclohexylurea. So this is the DCC, and this is not a phosphorus-based coupling agent. Now let me go to the next question, which says that which of the following derivatives is formed when DCC is used as a peptide coupling. Your options are number A, DNP derivative, number B, DCU derivative, number C, DCP derivative, and number D, DNS derivative. Which of the following derivatives is formed when DCC is used as a peptide coupling? I have shown you in the last slide what is DCC. Now, when DCC is used as a, as the particular coupling agent, what is formed as a side reagent? You may say as a side product. What is formed? DCC is converted to what? A DNP, DCU, DCP, or DNS. If DCC helps the coupling of the particular amino acids, the DCC will be converting to something. You have to identify what is that particular derivative. Again, I will wait for a minute, then I will show you the correct answer. Which of the following derivatives is formed when DCC is used as a peptide coupling? Let me check the chat box. Okay, I got one answer which is saying it is number D. Let me check if number D is the correct answer or not. So the correct answer will be number B, DCU derivative. So DCC method for peptide bond formation, I have already mentioned. DCC is the dicyclohexyl carbodiamide. Amide formation from the reaction of an amine carboxylic acid is slow, but that is why this reaction has to be accelerated by activating the carboxylic acid. So this is the DCC. What does this DCC do? It reacts with the carboxylic acid or the carboxylic acid functional group like this, and activates this to the amide bond formation. So that is how the amide bond formation happens. This activated acid now is susceptible enough to be attacked by amine, the carbonyl carbon group, and we have this amide bond formation, and we have this DCU formation, which is the dicyclohexyl urea, as the side products. Okay, this is the dicyclohexyl urea, and this is how the carboxylic acid is activated there is a O-acyl isourea intermediate is formed, and we have this our dicyclohexyl urea. Okay, that is how this thing happens, complete thing. Okay, so now we will go to the next problem. Problem number 17 says, which of the following statements regarding Merrifield resin is true? Number A, it is a solution phase peptide synthesis. Number B, it cannot be used to synthesize large peptide. Number C, here is a polymer bead is used, which is insoluble in solvent, which is used to solubilize the reagent. Number D, here T-Bock protection of amine is more significant than FMOC protection. Which of the following statements regarding Merrifield resin is true? Sorry. Which of the following statements regarding Merrifield resin is true? Number A, it is a solution phase peptide synthesis. Number B, it cannot be used to synthesize large size peptides. Number C, here polymer bead is used to which is insoluble in solvent, which is used to solubilize the reagent. Number D, here T-Bock protection of amine is more significant than FMOC protection. Identify the statement which is true regarding Merrifield resin. Which of the following statements regarding Merrifield resin is true? Let me check if there is any answer. Okay, I got one answer which is saying it is number C. Now let me check if number C is the correct answer or not. So the statement which is true regarding Merrifield resin is, yes, it is number C. Okay. So in case of solid phase peptide synthesis where the Merrifield resin is used, so it is a solid phase peptide synthesis, not the solution phase. So statement number 1 is not correct. Okay, it is false. Number B, it cannot be used to synthesize large size peptide. It is also false because it can be used. In fact, it is used to synthesize the large size peptides. Number C is correct. Here the polymer bead is used which is insoluble in the solvent, which is used to solubilize the reagent. Number D, here T-Bock protection of amine is not more significant. Okay, FMOC protection is actually the most significant, and it is the used in case of the solid phase peptide synthesis. So this is the structure of the Merrifield resin, as you can see. So solid phase peptide synthesis, starting material is bonded to the inert solid support, reactants are added in solution. Reaction occurs at the interface between the solid and solution because the starting material is bonded to the solid. Now, any product from the starting material remains bonded as well. Purification involves simply washing the byproducts from the solid support. So the first step is this is the resin linker. Okay, this is the functional resin bead, this is the linker, and this is the Merrifield resin. Okay, so the first step is the loading. So the loading of the first amino acid and also the protection of that amino acid using the FMOC protection group. Second step is the deprotection, adding base or solvent, base and solvent for the selective FMOC removal. Then we have this coupling of the incorporation of activated amino acid. So we have this activated amino acid which also has the N protected, and only C is carboxyl group is free, and the byproduct is formed is the DIC. It is the DIC oximopure protected. Okay. Then we have this coupling happens. Okay. Number three is the elongation, the repeating of the repetition of this step. That number third step is repeated for the elongation of the polypeptide chain. Then we have the deprotection, addition of base and solvent solution for the selective FMOC protection again. And the deprotection and cleavage. A TFA based cleavage cocktail is used to remove the particular growing polypeptide chain from this linker. Okay, and this is the resin linker from amide terminal sequence. So this is the resin linker. So this is the steps of the solid phase peptide synthesis. It is very easy. So first step you have to add the link the first amino acid to the resin bed, and then protect and deprotection chemistry also plays an important role in case of this solid phase peptide synthesis. Okay. Now I will go to the next problem. Problem number 18 says if chymotrypsin is used on the polypeptide chain, methionine, tyrosine, leucine, tryptophan, glycine, and phenylalanine, serine, how many fragments will be formed? If the chymotrypsin is used on the polypeptide chain, this polypeptide chain, how many fragments will be formed? Your options are number A 4, number B 3, number C 2, and number D 5. I already have mentioned in today's class itself what does chymotrypsin do, where chymotrypsin cleaves. Now you have to use that particular thing, you have to keep that thing in mind, and you have to answer that if the chymotrypsin is used on this particular polypeptide chain, how many fragments will be formed? Think, write in copy if it is necessary. It is very easy. I know all of you can answer it. I will wait for 2 minutes, then I will show you the correct answer. If chymotrypsin is used on the polypeptide chain, how many fragments will be formed? If chymotrypsin is used on this particular polypeptide chain, how many fragments will be formed? Let me check the chat box. Okay, there is no answer. So this is very easy. Actually, I have mentioned already earlier that how chymotrypsin works. How does it actually cleaves the particular polypeptide chain? Okay, let us check which of the following will be the correct answer. So if chymotrypsin is used on this particular polypeptide chain, how many fragments it will form? It will form 4 fragments. How? This is chymotrypsin. We all know chymotrypsin cleaves at the C-terminal end of an amino acid, which is aromatic. Okay, and provided the next amino acid is not proline. So we do not have proline in this sequence, so we do not have to worry about it. Now, Chymotrypsin breaks a polypeptide chain involving aromatic amino acid at the carboxylic end. About chymotrypsin, you have to remember that the adjacent amino acid, there is one restriction. If it is happens to be proline, that hydrolysis cannot take place. Okay, so this I have already mentioned, but in our given question, the particular chain, polypeptide chain, we do not have any proline. So we do not have to think about the restriction. Only we have to take care of how the chymotrypsin works and how it forms the cleavage. So this is our given polypeptide chain, the original protein chain. This is the N-terminal end. This is the C-terminal end. On the N-terminal, we have methionine. On the C-terminal, we have this particular serine. So chymotrypsin, it breaks the particular peptide bond on the C terminal of aromatic amino acid. What are the aromatic amino acids are there? Phenylalanine, tryptophan and tyrosine. So in this we have tyrosine, we have tryptophan, we also have phenylalanine. So chymotrypsin will break the C terminal bonds of these aromatic amino acids. So the on three places we will have the cleavage, this position and this position between tyrosine and leucine, between this and after tryptophan, and we have this between the phenylalanine and serine. Okay, so and we have these are our fragments. This is the original N terminal and new C terminal. Then it is the then this fragment, and this fragment. So we have 1, 2, and 3, three dipeptides and one single serine amino acid. So these are the different fragments which are formed if this polypeptide is used, if chymotrypsin is used on this particular polypeptide. Okay, so that's how. If this kind of question is given to you, you have to solve it by doing this kind of. You have to think and you have to write, and then you can easily answer this kind of question. Okay, now let me go to the next problem, problem number 19, which says which of the following correct consequence of a hexapeptide based on the following statement. First statement is amino acid composition. We have two R, means two arginines, one A alanine, one serine, one valine, and one Y, means tyrosine. Okay, Sanger's reagent give us the DNPA derivative. Trypsin digest give us two peptides RAV and RSY. Chymotrypsin digest give us two peptide ARVY and RS. So what will be the correct sequence of the hexapeptide? Number A, AYVRSR. Number B, AVRYSR. Number C, AVRYRS. And number D, AVAYVRRS. Which of the following will be the correct sequence of the hexapeptide based on these statements? Read the statements very carefully. I will give you 2 minutes. Try to answer this question. This is a little bit tricky, I think, but you can easily answer this if you read the question statements carefully. Let me wait for 2 minutes, then I will show you the correct answer, and I will also show you how to solve this kind of question. Which of the following will be the correct sequence of the hexapeptide? So, is there any answer in the chat box? Let me check. Okay, I got one answer which saying it is number C. Let me check if number C is the correct answer or not. Number B is actually the correct answer. Let me show you why. So basically, let me just use the pen. So now let us say check the statements. First statement is the amino acid composition is this one is fine. Sanger's reagent gives us this DNP derivative, this fine. We don't have any much information from this two because all the hexapeptides which are given in option have this A in the N terminal, so all will form the DNPA. Trypsin digest give us the two form fragments. Now trypsin digest, what does trypsin do? Trypsin basically cleaves the peptide on the C terminal of the lysine and arginine. We do not have any lysine here, we only have arginine, which is R. So now let us cut on the left side of the arginines. Okay, so if on the first hexapeptide, if it cuts on the C terminal, it will give us these two fragments, but it does not match with this third statement, so it is not that particular answer, correct answer. Number B, if we cut on the carbon this side after the arginine, the C terminal of the arginine, it gives us two fragments RAV, RA and V, RSY, RSY. This matches the criteria number 3. If this for this number C, if we cleaves at this and this position will be cleaved, we will have three fragment. It also does not match the this statement number 3. And in case of number D also, if we cut here and here, it does not match. Only this particular step number B only matches with the statement number C, right? And number fourth statement, which says the chymotrypsin digest will give us these two amino acids. Now, if chymotrypsin digest happens, okay, let me just check the color of the pen. If chymotrypsin digest happen, chymotrypsin, where does it cleaves? It cleaves the C terminal of the aromatic amino acid. Aromatic amino acid, which is only this tyrosine. If so, if it cleaves here, we get this fragment AVRY, ARVY, and RS. This statement. So only this, it also matches with this number statement number 4. So only our this hexapeptide, which is number B, matches our all criteria. That is why this is the correct answer. That's how you have to solve, or you have to do this type of question. You have to take each and every peptide. Okay, whatever the answer is given in option, you have to take each and every peptide and you have to check if it is matching all the criteria that are given in the statements in the question. Okay, so this was the correct answer. Now the last question of today's session, it says that the hydrogen bonding interaction is the primary stabilizing force of the dash structure of the protein. Your options are number A alpha helix, number B beta pleated sheet, number C both, and number D none. Hydrogen bonding interaction is the primary stabilizing force of dash structure of the protein. Which will be the correct answer? This is the last question of today's session. Your options are number A alpha helix, number B beta pleated sheet, number C both, and number D is the none. Hydrogen bonding interaction is the primary stabilizing force of which structure of the protein? Hydrogen bonding interaction is the primary stabilizing force of the dash structure of protein. Let me check the chat box. This one is the last question of today's session. Let me check if there is any answer for the last question. Okay, I don't think there is any answer. Okay, let us check what is the correct answer of the last question of today's session. So, hydrogen bonding interaction is the primary stabilizing force for the both alpha helix and beta sheet. So basically These are the secondary structures of the protein, the alpha helix and beta pleated sheets. Okay, the secondary structure of the protein consists of the regions of ordered structure adopted by the protein chain. In structural proteins such as wool and silk, secondary structures are extensive and determine the overall shape of the properties of the protein. However, there are also regions of the secondary structure where most other proteins. There are three main secondary structure: alpha helix, beta pleated sheet and beta turn. Alpha helix, the alpha helix results from the coiling of the protein chain in such a the way that the peptide bonds making up the backbone are able to form the hydrogen bonds between each other. These hydrogen bonds are directed along the axis of the helix as shown in this figure number 2.5. The side chains are component of the component amino acids stick out at the angle right angle from the axis of the helix and thus minimizing the steric interaction. Further stabilizing structure are the less common type of helices are the 310 helix, which is more stretched from the ideal alpha helix, and the pi helix, which is more compact and is extremely rare. So this is the alpha helix, as you can see. Beta, and here also you can see how the hydrogen bonding is marked here in the blue color. The beta pleated sheet is like this one, are the layering of the protein chain one on the top of the other. Okay, here too the structure is held together by hydrogen bonds between the peptide chains, and the side chains are situated at the right angles to the sheets. Once again, reducing the steric interaction, the chains of the beta sheets can run opposite direction, anti-parallel or the same direction parallel. So these are the beta pleated sheet, and the side chains are sticked above or the below the plane of this particular sheet. Okay, so these are the secondary structures of the protein. So this was the last question of today's session. This particular question, the secondary structures, we will discuss in detail in our next class, which is our week 3 live session. So I will share my email ID here in the chat box. If you have any doubt or any query, please feel free to drop an email to this email ID, or you can also post your queries in the discussion forum of the particular page, okay, which is present in the SWAYAM portal. You will find there is also a discussion forum, so you can also write your course problem. You will find the problem solving session recording and the PPT here. I have already shown you at the beginning of the class. So if you have any doubt, any query, feel free to reach me through this email. Okay, I will try to answer your queries in the next class. So today it was our week second live session of this organic chemistry in biology and drug development. We extensively discussed about the different reagent to form the peptide bond. We different protecting, deprotecting reagents we used to we discussed the reagent to identify the or the particularly the for the cleavages, how the cleavages happen. We discussed about the amine protection, carboxylic acid protection, peptide synthesis also. We discussed a little bit about the solid phase peptide synthesis, and we discussed about the the coupling reagents also. Last we discussed about the secondary structure, and in detail we will discuss it in the next class. So thank you for joining. I will close the session for today. If there is any no doubt or or there is no query, our next class will be on Friday 8th August from 6 p.m. Okay, kindly join the class, and we will discuss week 3 course content on that day. So I will close the session for today. Please let me know if there is any issue or there is any doubt in the next class, or you can drop an email to this email ID. Thank you so much for joining. Have a good day.